Tēnā koutou katoa, ko te amana te whakapapana mai, nāre, ko kirikiri o te whenua tapu, ko Bridgette Farnworth, I heard tēnā tātou katoa. Hi everybody, my name is Bridgette Farnworth and I'm currently aligned with Malcolm Marlborough Institute of Technology, where I work as a researcher. So I'm a wildlife biologist and it's my pleasure to be invited today to talk about the tiny teaspoon of knowledge that I have on some of the ecological impacts that light pollution can have. So, as a wildlife biologist, one of the questions that we sort of start asking ourselves is, about these emerging conservation threats is, well, is light pollution, is this actually a problem for New Zealand? And I did this and I published a paper last year where we took some of the Starlight data and did some modelling, and we found that the extent of light at night is increasing in New Zealand on average at a rate of 3.2% per annum. Now that doesn't sound like a lot, until we compare it to the global average, which is 2.2%. So, yes, it is a problem for New Zealand, and particularly for our nocturnal species, because that's the next question that we ask ourselves. Well, if it is a problem, how does it affect our ecology? And do we have any evidence of some of those impacts? So, within that same paper, what we did was we looked at all of the available information that we have in New Zealand on our ecology, and what we found was a little bit surprising. So we don't actually have that much information. There are 39 pieces of information, and when we talk about information, what we're talking about are theses that have come from universities, peer-reviewed published papers, or reports that have come from perhaps some of our Crown Research Institutes, or from consultancies as well. What this data showed us was that we have a variety of species that are looked at, and definitely the information is dominated by birds. So 17 pieces of information about birds, 12 to do with mammals, 9 for invertebrates, 4 for fish, and only 1 study that talks about plants. When we look at some of these impacts, we can sort of summarise these in a big picture. Again, thanks to Ellen and her fabulous work on this. This is a graphic that was produced as a part of the research, and I'm just going to talk about some of the impacts that we see throughout the literature, just with some examples. So if we look at the first example that we can see is phenology and how light changes this. So phenology is our ecological geek term for the timing of bud burst and flowering for plants. And this was an observation that was made where the Bridgette tree, which is New Zealand's Christmas tree, was flowering more profusely under street lighting than its neighbourhood counterpart that was in the darkness. We can also see how communication is impacted by species. So in this study, it was a really nice work that was done by Ellery McNaughton and colleagues from the University of Auckland, and they had the opportunity to collect some acoustic data from an area that was experiencing a retrofit. So the street lights were moving from HBS lighting to LED lighting, and so they put out some acoustic recorders, and we could see how the species changed their calling in response to the presence of light. So they had the tui, which is the bird over your left, and the invasive minor bird on the right-hand side, and they found that both of these species started calling a little bit later. There was no impact on the end of their calling at the end of the day though. Now tui were very consistent in this, minor were very consistent in this response. Tui, this only occurred across some months, so there might have been some other variables in there as well. We can see how navigation can be impacted, and this is particularly strong for our seabirds. So New Zealand seabirds, unfortunately, they tend to get a little bit distracted by lighting. I think someone else is going to give a talk about seabird lighting, so all I'm going to say is that they can experience something that's called fallout. And this is where we have birds that, particularly the fledglings, as they're taking off from their burrows, they get a bit distracted, disorientated by lights, and they crash land, and they cannot get up again without help. What we see as a result of this is some work that's come out by Ariel Heswell, again from the Auckland region, where the Cook's petrel was studied, and some of the birds that had crash landed and had been taken to a wildlife rehabilitation facility and cared for there, unfortunately they died. So they had 17 of those birds, 70% of those had and the necropsies that were performed on them, they were found to have sustained internal injuries and sort of head trauma that was consistent with sort of crash landing because of this fallout. So these birds were otherwise healthy. So what we are starting to see here is some of the effects that can be attributed to the result of light at night influencing navigation. We can also look at positive phototaxis. So that's where we have attraction that occurs towards light. And this is really well known for invertebrates. And the study I'm going to talk about is Jess Schofield's master's thesis, which came from the University of Canterbury. And this looked at caddisfly attraction towards the light. And what they found was they tested lights in two different areas, so light naive areas and in urban areas. And they also tested two different colour temperatures. So one that was slightly warmer and one that was slightly cooler. And what they found was that caddisfly attraction was greater, it was equivalent in both of the areas, but it was greater towards the cooler LED lighting. And finally, we can talk about negative phototaxis. And this is not as common, not as commonly studied, but it's when we have, I guess, species that are moving away from the lighting. And I'm going to talk about this in general because this is an area that I work in. And the first example I'm going to talk about is New Zealand's ugliest bug, which is highlighted here, and that is the weta. So, do these species avoid the light? That's the next question we're asking, and we're looking at the Auckland tree weta as an example. So this project was undertaken at Ngātautari Ecological Island, which is the largest pest fence reserve in New Zealand. And I'm aware that we have some international guests here, so just to explain, this is like a forest that has a very large fence surrounding it. It's 2 metres high, it has a specialised mesh to prevent mammalian predators from entering into the reserve, and it has a hood feature on top to stop climbing mammals such as possums from making their way in. The purpose of it is to protect our avifauna, which experience very negative consequences from predation. The benefit of it though is the invertebrates are also doing really well here, which is why we could look at weta and we could use a section of the fence that runs through the reserve to examine this question. So I set up 15 stations at Ngātautari. We had a little magnetic light that we could pop up into the hood, and we had a trail camera. So trail cameras can take pictures at night at various intervals. And what we managed to capture was some really cool shots of the weta roaming around inside the hood. So again, just for our international guests, weta are some of the largest flightless orthopterans that we have in the world. They're like a huge, ugly cricket, basically. So we're looking at Ngātautari, and there are two different species that we were observing. So we had cave weta, 246 observations of cave weta, and 315 observations of tree weta. And so then we could look at how light influenced their presence. And what we found is that they are quite strongly affected. So this graph is showing the average number of observations in light. That's on the y-axis. The x-axis is our species, and yellow is presence during illumination, grey is presence during the dark. And you can see the effect very, very clearly. Both of them were found significantly more often in the dark than in the light. So light is materially species. The really cool thing about Auckland tree weta is that they are what we call sexually dimorphic. So the males and females look different from one another. So the females, if you want to call it that, have a tiny, cute face and this larger body. The males, on the other hand, have these huge, enormous faces, and they're kind of shaped like an upside-down light bulb. So it's quite easy to tell the difference between them in those photos. We could then look at how sex was affected by lighting treatment. And what we found were that males, again, grey being dark, yellow being light, males were observed significantly more often in the dark than in the light. Females, however, we did not detect a difference for. So here we have a species that is showing a sexual differentiation in behaviour in response to the presence of light. And I can hear what you're all thinking, which is, well, this is New Zealand. We have an entire country that's full of weird and wonderful creatures. So perhaps an ugly grasshopper that avoids the light, it's not that much of a big deal. And that's why I wanted to talk about the second species, which is probably the world's most well-known invasive species, the ship rat. The ship rat is also known as the black rat or roof rat overseas. It's all the same species. That's Rattus rattus. So for this study, it was performed down at Lincoln, and we brought 20 wild ship rats into captivity. And anybody who has worked with ship rats before, that's not very easy. So we brought these individuals in, and we worked in this enormous pen. So it had 3 separate chambers, or 3 chambers within this enormous pen. And that middle chamber was configured to operate like a U-maze. So if they wanted to explore the entirety of the pen, they needed to make their way through the U-maze. Now that U-maze could experience 2 different conditions. So we had dark, and we also had light. And we had some cameras within the middle of the pen so we could observe their movement behaviour. So this is what it looked like. Here we have a rat, and he's making his way out from the sort of entrance part of that middle chamber through to the other side, and here he comes again. Now when we turn the lights on, I think you'll notice an immediate difference. That movement behaviour is a lot slower. I think what you might also be able to see is that there's sort of a rectangle that is tracking the individual moving around. So that's some tracking software that was developed by a colleague that I worked with, and what it enabled us to do was to track the rate of pixel change, and so that we could use as a proxy for speed. So essentially, now in these graphs that I'm showing you, on the left-hand side there, rate of movement, that's what we were able to determine. So we can see speed was significantly affected, and they are moving a lot slower in the light than in the dark. We can also see the number of times that they would visit that middle chamber was also reduced after we had turned the lights on. So the middle graph there is showing that significantly fewer visits made in the light. And finally, the third graph here on the far right-hand side is showing the number of crossings that they were willing to make. So entering from one side, moving all the way around to the other side. Again, significantly reduced under illumination, which again are the yellow bars, the grey bars, and the dark. Okay, why are they avoiding the light? One of the reasons that we've put forward for this has to do with the evolutionary relationship that they have with predation risk. So essentially, when these animals are moving about in time and space, not all areas are risky and not all areas are safe. It's the same thing. So essentially they need to be able to determine when it is safe for them to be out and about undertaking these activities. And one of the things that we notice about these prey species, such as rats and weta, is that they use these environmental cues to be able to tell them this information about when it's safe for them and when it's risky. Ambient light, such as moonlight, is used as a cue of risk. So essentially, these species are taking in this environmental information, and during these periods of high risk, such as when the moon is out, they will avoid feeding or moving or performing their other activities. And when that risk subsides, they'll resume them. And we see this from the literature, even just with the influence of moonlight. It's particularly strong for rodents. Okay. So, just to recap, we've got individuals that will change their behaviour in response to lighting. We also understand that this is because some of those predation dynamics will also change. So, why does this change matter? Why do we actually care if things are maybe avoiding the light or being attracted to light? And that's what I'm in the middle of studying at the moment. So I just wanted to share a bit about this. So I'm working with invertebrate communities, and when we talk about invertebrate communities, basically we're talking about all of the species that are present within a particular area and how they interact with the other species that are there. And invertebrates are a really great group to work with because 60% of them are nocturnal, and so they're very sensitive to some of these changes in ambient light levels. Invertebrates are also responsible for a lot of our really vital ecosystem services. So the things that we basically get for free from nature that benefit us as humans. And these include things like pollination, like we've already heard, but also biocontrol. So that's where we have predatory invertebrates that keep the prey species in check so they don't blow up out of proportion. I mean, one fly in the room is annoying, imagine 100 of them, right? And the value of that role that they have there, I think in the US alone, this is an old statistic, but it was like 3.3 billion. So they're performing really, really important services for us. One of the things that we can see when light sort of influences these communities are that in the dark, we have these regular proportions of predator and prey species. But when we turn the lights on, internationally what we're seeing are that more predators are attracted towards the light. That's one response we could see. And on the other hand, we're having fewer of the prey species that are present within the area. So I wanted to see if this also occurred within a New Zealand setting. And so I've been back out at Mangatotiri Ecological Island to perform an experiment there. And so I had 12 of these stations that were set up with lights out in the middle of nowhere to do this. So we had 12 that were in the dark to collect some baseline data, and then we turned half of them on, and we could have a period where they were before and after, so we were able to collect that data and look at it in a really robust way. Now at each of these stations we had pitfall traps, and these are used to collect ground-based invertebrates. So a pitfall trap is essentially a cup that's dug into the ground, and it has a level of preservative fluid in it. So anything that walks along will fall into the cup and it dies. And that makes me feel like a terrible conservation biologist, but we are able to have this huge collection of species that are present, so we can look at the types of species that are present and the number of them that are there as well. So in these pictures you can see some carabid beetles on your left-hand side there, some decent spiders, I think, and some of those cave weta again that are showing up in these traps. We've just started to get the data in for this, so I thought I'd just give a real preliminary analysis of this. Okay, so I'm going to roll over this graph in a second, but just so you know what we're talking about, we've got the species groups along there, and then your abundance at the bottom, and these bars, dark this time represents things that are out at night, green represents things that are out during the day. And I just wanted to firstly talk about some of these nocturnal rhythms, and that it should be a little bit obvious that what we are seeing in forest communities is that everything comes out at night, perhaps with the exception of diaphragm wasps. So these guys tend to be a little more diurnal. So diaphragm wasps, these are parasitoid wasps. They're probably out because they're looking for some kind of grub that they can pick up, lay their eggs into so their babies have something to eat. Otherwise, we've got centipedes, they are nocturnal, decid spiders. The bulk of these I think are Bridgette, which is a web building spider essentially. Huge big jaws on the front, really long legs. Rogue beetles, they're about 50-50, daytime and nighttime. Could be some species specific nuances there. Landhoppers, nocturnal. Cave weta, very strongly nocturnal. Millipedes, nocturnal. Tenonathus, this is a species of carabid beetle, so they are predatory. I think you might be able to see the jaws at the front of it there that eat things. Mites and ticks are also nocturnal as well. So if we look at some of those key groups in here, when we add the lights, we can sort of start to get a bit of an idea about what is happening. So this time the graph is showing the dark bars. This is just the things that are out at night during that nighttime period. This time the grey bars are when the lights are off, yellow bars when the lights are on. So cave weta, we are seeing fewer of them when the lights are turned on. Also I should mention, these are just averages. I have not run statistics on them. So Tenonathus, very, very strong response to light. Landhoppers, again, another response to light there, where there are fewer of them within the community. Millipedes and any of the diaphragm wasps that did happen to be out at night, they actually seem to be about 50-50. They don't seem to have too much of a response. Millipedes, I think this could be because a lot of our species apparently don't have any eyes. So I'm not sure how they are sensing what their perception mechanism for light is in the first place. Decid spiders might be the only ones that are actually attracted to lights, but when we think about it, those predation dynamics again, these guys are web builders, their prey is likely to be attracted to the light, so it makes sense for them to build their webs in places where they're going to receive more food. So that's what we have at the moment, and I'm looking forward to getting further into it as we progress with the work. Okay, so why does this matter? Why does it matter if we're seeing these changes in community structure and in the dynamics? And for me, this is the interaction that really matters. So if we are having more prey and more predators within a system, but their prey is changing, what does that actually mean for the things that they eat? What does that mean for the ecosystem service of biocontrol, so their ability to keep annoying insects in check for us? What does that mean? If you think about it, we might be sort of changing the availability of proportions of food that are actually really good for them. So I think about it as in, this might be like you being at a buffet and you've got salads, vegetables, and then there's the dessert carts. You can eat things in proportion. Imagine if then you turn the lights on, suddenly only the dessert part is available. Now dessert is delicious, but it may not necessarily be as nutritionally balanced for you as if you had the whole smorgasbord there. And it's a really, really tricky question to answer, but I'm doing my best with it. I'm right in the middle of it at the moment. This is the banded tunnel web spider, which I'm working on to address this question. This is New Zealand's heaviest spider. I don't think it's the largest, but it is New Zealand's heaviest. I just think it's New Zealand's hairiest and scariest spider, but it's been a real joy to work with it. Fortunately, it's dead in this picture, but what we've been able to do with this is to do gut content analysis, so to look at what they're eating, and we can do this genetically, and we've also talked to various experts in the field about what we think that they will be eating. And what we're finding through eDNA is that they will eat millipedes, so the furry snake millipede, and they also eat those carabid beetles that we've been talking about. Now the interesting thing about this, if you remember from our graphs, is that when we turn the lights on, the millipedes are still there. Ctenonaphis is no longer within the system. So what we'll be able to do shortly is to do a nutritional analysis to see, are these things still occurring in their balanced proportions, and are they as nutritionally good for the tunnel web spider as what they need. So can we mitigate these impacts? I don't think you're going to like the answer to this because we don't have enough information to be able to, I think, put forward appropriate mitigatory measures at the moment. So this is from the study that we did where we looked at all of the impacts at light at night from New Zealand literature. This graph is a summary of the types of responses that we have data on at the moment. And you can see it's definitely dominated by phototaxis. That's the most amount of information that we have. Things like reproduction, population size, some of those community composition. We've only got a handful of studies about it. So what we need to do is increase the range of responses that we are looking at within our ecological studies. In addition to that, coming back to this graph again, what we can see here are that there are entire taxonomic groups that are missing and that we don't have information about. So reptiles and lizards, for example, we have no studies about the impact of artificial light at night on that particular group. The same for marine mammals. So we have to broaden the range of taxa that we are also working with. And so we need this information because we cannot mitigate what we do not understand. We have to have the information first to be able to collaborate with all of the various partners to say, here are some suggestions for what we could do moving forward. And so I'd just like to thank you for the opportunity to be here to speak and for partners that we worked with in the funding contributions. I'd especially like to thank my co-author and colleague Ellen. Ellen has been absolutely instrumental to this work, and it's not very often I get to publicly embarrass her with my gratitude. So thank you, Ellen, for your support for this work. And I will finish and take your questions. Thank you.
Great, thank you very much for that, Bridgette, and there's plenty of time for questions, so thank you for being so timely. Let's start at the front here, just because the mic's here, and then we'll move to.
Thank you so much. It's fascinating to see this fundamental basic research, and I can't quite believe you're studying what these insects have been eating for lunch. It's just mind-boggling what scientists do these days. I want to come back to kind of where you ended about the challenge, like we don't know enough to mitigate. I kind of want to maybe challenge you a little bit on that and have you kind of help us think through that. We've also heard that every species responds differently to different spectrum, but one of the constants is the minute you shine light on something, it's going to have an adverse impact. It's going to affect something. Mm-hmm. So I think part of the challenge is like, how can we continue to support the work that's needed to establish these thresholds, and also not allow policymakers say, oh, we don't know how to mitigate it, therefore we don't need to take action, because the mitigation, when you get down to it, once we got to it, they're going to be the same. Put the light where you need it, dim it down when you can, turn it off when it's not, and yes, we'd always like to know more. So I'm curious, like, how we should be thinking about that, and again, how we can ensure that we don't allow the policymakers a sort of get out of jail card.
No, absolutely. And I agree with your point. I have probably summarised that a little too succinctly when I say we can't mitigate what we don't understand. I'm probably coming at that from an academic viewpoint of when we have to put forward these research proposals to be able to attract funding to work in this area. That's probably my angle for it. I think you're right though, where you can almost guarantee if you're putting a light in the space, something ecologically in there is not going to be happy. My point for saying we need more information about it is globally the responses that we've seen from organisms are that there are these behavioural responses to light. And I think moving on from that, I think the challenge is to say, well, what are the consequences of that attraction or repulsion by light? And I think that might be what sort of helps us get in the door to make these sort of stronger policy movements. I think about it like what happens, if you've got a line of dominoes and we're seeing what happens when we flick off the first domino, and I think we need to know sort of what happens the fifth, sixth, seventh down the chain as well to have some really strong conversations about that.
Thank you, that was fascinating. I had a question about your shipwreck research and wondering about whether it can be applied in the predator space where, particularly with mohua and bats, where you know bursting trees, you thought that there's a potential application, and I'd just like to hear your thoughts around using light as a way of keeping rats away, particularly during nesting seasons.
Yeah, that's actually was one of the original objectives of the work that we were taking, and I think I don't know if this is a popular place to say this, but the thought was New Zealand has what we call peninsula sanctuaries in addition to our ring-fenced one. At peninsula sanctuaries, they can fence the entire way across the sanctuary, but when it reaches the edge of the ocean, you don't have the physical structure to be able to sort of permit into the ocean. And you can't put traps there because of the tide coming in and out. That's going to obviously damage these physical structures, but also damage any traps that you put there. What happens then is you have an entire period where the tide is out, where you have big exposed areas that predators can make their way around into these essentially pest fence reserves. So there was some talk about, is it an option to carefully controlled areas instead of having to put out chemical deterrents or traps or that sort of thing? Is that an option? Which is sort of what kind of got the work kicked off to begin with. Whether it would actually work or not, and whether it's actually a good idea in the context of light pollution, I think is another question. Because what we see with some of these, essentially we're dealing with predation cues of risk. What we see is sometimes we can have organisms that habituate, so they get used to it. And also at the end of the day, if you're hungry and you want to be out and about, it's, can I die because I'm going to starve, or I can die from a predator, which risk are you going to sort of take? So how effective it would actually be is a different question.
I think so there's also been some talk to use it as an extra precaution if fences go down.
Yes,
right?
that's true. Yes.
Application, has that been put into place?
No, that was the other conversation that we were having about it, which is where at these pest fence reserves, obviously significant time and money has been put into keeping them pest free. What happens when we have storms is that we have tree fall, it will damage the fence, and so there was again some conversations about could we use it as a short-term deterrent to keep this mainly rodents away, because we know from existing research that the first thing that will encounter a breach in a fence will be a rodent that will be within the first hour, and there's a 99% probability of that occurring.
Thanks a lot for your presentation. What I wonder is just more a technical thing about the traps that you were using. So it was a kind of passive method, or were the insects somehow attracted to the trap?
So the preservative that we were using was monopropylene glycol. There have been some studies that have indicated that there can be attraction to ethanol by some particular species. To my knowledge, monopropylene glycol is a very commonly used preservative. I don't think the attraction factor is as strong for it, but we weren't baiting our traps.
Okay, so there is no bias of species that are more attracted to those to the traps because there's no attraction.
Should it be, to my knowledge? Yeah.
Okay, thank you.
You're welcome.
Right, maybe one last question. Maybe Elizabeth, if you can.
Hi, just quickly, I was just wondering what the colour temperature of the lights you used in your experiments was.
In the existing ones. So they were warm white light, 2,700 degrees Kelvin, and the lux level at the ground, they were sort of set to replicate roughly what street light would be, so 10 minutes at the ground. So the thought was to make it as applied as close as possible to those implementations.
So, thank you.
Great. Please join me in thanking Bridgette.